Today we're going to talk about the basics of the life chemistry, and we're going to start with a conversation about the elements that constitute the majority of the living things. There are 5 elements, which are carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus, that can be found in virtually all living tissues. Carbon, oxygen, and hydrogen will be present in all biological molecules. Nitrogen can be found in DNA, RNA, and proteins. Sulfur is in proteins, and phosphorus in DNA, RNA, proteins, and phospholipids. In small quantities, calcium, sodium, chlorine, potassium, and magnesium, these ions, can be found in a human organism. They're responsible for regulation of enzymatic activities, permeability of the membranes, and ionic balance. Some elements are found in the trace quantities, such as iron, iodine, selenium, molybdenum, and copper, and many others. So, in order to understand how biological macromolecules function, we need to first address some questions about chemistry in general. All chemical elements consist of atoms. It's the smallest unit that is chemically indivisible. This unit represents a particular element. When atoms react with other atoms, they form compounds and/or molecules. Atom consists of a central nucleus and the electrons that orbit the nucleus. Nucleus consists of the protons that are positively charged and neutrons that bear no charge at all. Electrons that are charged negatively, right here, orbit the nucleus, and these negatively charged electrons are attracted to the protons. And electrons do not fly out of the atom because they are attracted, electromagnetically attracted to the protons. So obviously, since atom is electrically neutral, the number of electrons is equal to the number of protons. I want to highlight that the mass of an electron is very small, and the majority of mass that constitutes the atom I'm sorry. The majority of the mass that constitutes the atom, the overwhelming, almost entirely, the atomic mass, is concentrated in the nucleus. So it's protons and neutrons that are heavy. And the masses of neutrons and protons are practically the same. Now, electrons orbit the nucleus along the circular, more or less, well, it's not circle, but let's put them in the kind of lines that we call orbitals. They're not exactly lines, but, different orbitals, shown here, have different energy levels, and the energy of an orbital increases as the distance from the nucleus for this orbital increases. So orbital number 3 will have higher energy than orbital number 1. So the electrons that are farther from the nucleus will have higher energy levels. The outermost energy level, the outermost shell, in this example it's a number 3, is called a valence shell. Valence electrons have the highest energy, and they are the easiest to separate from the atom. If electrons are separated from the atom loses some of its negative charge and becomes itself, it was neutral, remember it was neutral, but when it loses some of the electrical charge, some of the electron charge, it becomes a charged ion. So how do we identify atoms? We identify them by a number and a mass and a symbol. So chemical symbol is this letter: C for carbon, O for oxygen, S for sulfur, and so on. On top of that symbol right here, there is an atomic number, which represents the number of protons in the nucleus. Remember that it's important to understand number of protons, not necessarily electrons, because if electrons leave, you end up with an ion, but atomic number, it's the number of protons in the nucleus. And this is a really important. All atoms of the element have the same atomic number. Okay? So no matter which atom of carbon you take, it's going to have 6 protons in the nucleus, and its atomic number will be 6. A mass number, or atomic mass, is the total number of protons and neutrons combined. You see? So mass of each proton or neutron, as we mentioned, they are virtually equal. It's called 1 atomic mass unit, and we write this above carbon. This picture here isn't entirely correct. So now think of it this way: if the atomic number uniquely characterizes an element, and all atoms of the same element have the same number of protons, means that you can add or take away neutrons and it will still remain the same element. A few examples are here. I show you 3 isotopes of a carbon. So isotope of an element has the same number of protons, it's always 6, but different number of neutrons. Isotopes are chemically identical, but can be physically different. Okay? So here you can see isotope of carbon with the atomic number 6 but atomic mass 12, isotope of carbon with atomic mass 13 and 14. So lighter, and 14 is heavier. Same goes for phosphorus. All isotopes of phosphorus have 15 protons but may have different number of neutrons. For instance, phosphorus 31 is a stable isotope, but phosphorus 32 and phosphorus 33 are radioactive isotopes that undergo a radioactive decay called beta radiation. So the importance of this feature that some isotopes are stable and other isotopes are not, and then can release radioactive energy, it's really important for modern medicine, because radioactive isotopes can be used in the treatment of cancer or labeling certain structures inside the human body, or creating a gamma ray that can be used to eradicate cancerous cells. Elements that we know are organized in what is called a periodic table of elements. It's a logical pattern consisting of horizontal periods and vertical groups. Elements are organized according to their atomic number. Atomic number of hydrogen is 1, helium is 2, lithium 3, beryllium 4, and so on and so forth. So why do we arrange elements in this particular way? Because the number of protons in the nucleus, or atomic number, corresponds to the number of electrons and corresponds to reactivity of the element, its ability to react with other elements, forming molecules and or compounds. So what is common between the elements in the same column, the same group? Now, if you look at the group 1, which contains hydrogen, lithium, sodium, and then down there's potassium, you will notice that all of those have one valent electron. Look at the group number 14. In the group number 14, carbon and silicon have 4 valent electrons, 4 electrons on the outer shell. 17, 2, 4, 6, 7 valence electron. 18, 2, 2, 2, 2, 8 total. So basically, elements in the same column, the same group, the same number of valence electrons, and they all have similar reactive properties. Now, elements in the same period, let me erase this. Now, elements in the same period have same number of orbitals. 1 for period 1, 2 for period 2, 1, 2, 3 for period 3. This will also determine how reactive the valence electrons are. For instance, valence electrons of lithium, if we go back one, valence electrons of, well, it's sodium, since they are more reactive than valence electrons of magnesium. When atoms combine together, they form chemical bonds. And these bonds determine the reactions between various chemical compounds, how these compounds are being formed, being broken down, and being reformed. There are 3 types of chemical bonds: ionic, covalent, and nonpolar covalent. So which electrons form bonds? If you would look at the valence shells, the outermost shells of the elements, you will see that they may contain up to 8 electrons, with one exception being the first period. Hydrogen and helium, on the outer shell, they can have no more than 2 electrons. Let's go back here. So atoms with the filled valence shells, atoms that contain 8 electrons on their valence shells, are the most stable, and they do not form bonds with other, that would be noble gases like argon or neon. Atoms that have their valence shells unfilled, they can form chemical bonds, obviously. Now, if you would look at the periodic table, you will see that on the left there are elements with 1, 2, or maybe 3 electrons on their valence shells, while on the right you have elements with 6, 7 electrons on their valence shells. So atoms with 1, 2, 3 electrons are more likely to lose electrons or to donate electrons to atoms that have 5 to 7 electrons. Okay, so sodium is likely to donate its electron right here. Sodium has only 1 electron on its valence shell, to chlorine, which has 7 electrons on its valence shell. When atom loses an electron, it forms a positively charged ion, cation, or if it donates electron, it forms a polar bond, and it has fewer electrons and has less negative charge. If the atom accepts electrons, gains electrons, so we're talking atoms with 5, 6, or 7 electrons, it forms a negatively charged ion, anion, and again, a polar bond. So when atoms combine, 2 or more atoms combine, they form a molecule. Now, hydrogen, for instance, is a molecule that is formed by 2 identical atoms. Okay? A compound is also a molecule, but it has to contain different kinds of atoms. For example, carbon dioxide or water are compounds. So hydrogen is not a compound, but a molecule. CO2 is both a molecule and a compound. Let's talk about different types of bonds. Ionic bonds are formed between positive ion, the cation, and negative ion, anion. When, in this example, sodium atom, having 1 valence electron, loses it, this electron is being accepted by the chlorine atom, which has 7 valence electrons. Thus, sodium becomes positively charged, a cation, and chlorine becomes negatively charged anion. This oppositely charged sodium and chlorine ions are attracted to each other. Okay? Now, what about a covalent bond? So covalent bond can be polar or non-polar. The fundamental feature, the covalent bond, is that atoms do not exchange electrons, they share valence electrons. So in the end, each atom fills its valence shell. And covalent bonds are important and very common in the compounds that comprise the living tissue. So let's take a look at nonpolar covalent bond first. So nonpolar covalent bond, the electrons are equally shared between 2 atoms. For instance, in a molecule of methane, the electrons are equally shared between the atom of carbon and atoms of hydrogen. Okay? When there is 1 pair of valence electrons is shared, like in a molecule of methane or in a molecule of hydrogen, that will constitute a single bond. When 2 atoms share 2 pairs of valence electrons, like in a molecule of oxygen, that'll be a double bond. And if 3 pairs of valent electrons are shared, like in an molecule of nitrogen, that will be a triple bond. Okay. Usually, nonpolar covalent bonds are formed between the same atoms. There are some exceptions, like here, a molecule of methane, and in general, bonds between the carbon and hydrogen are nonpolar, but mostly nonpolar covalent bonds are formed between the same atoms. Now, polar covalent bonds. In this case, 1 atom attracts electrons stronger than another. For instance, in the molecule of water, oxygen has more protons in its nucleus, more positively charged protons in its nucleus than hydrogens. So it attracts electrons stronger, and it sort of gains bigger share of those shared electrons. Electrons sort of shift towards oxygen, which gives it partially negative charge and gives hydrogen partially positive charge. But another example is carbon dioxide. Oxygen, having more protons than carbon in its nucleus, shifts the electrons to itself, gaining partially negative charge and leaving carbon with partially positive charge. Now, if you look at the molecule that is somewhat similar, carbon dioxide, in this case, there is no shift in electrons to oxygen to carbon, because oxygens on both sides counterbalance each other. So in this case, bonds are not polar. Okay? Sorry, not bonds. The molecule is non-polar. Basically, in the polar covalent bond, electrons orbit one atom more than they orbit another. So let's take a look at the molecule of water. In a molecule of water, as we mentioned, there is a slight negative charge at oxygen, slight positive charge at hydrogen. So what does that mean? That means that this hydrogen and this oxygen can form electromagnetic bond, which is called a hydrogen bond. So as we mentioned, since electrons orbit oxygen more than they orbit hydrogen, there's a polarity in the molecule. Molecule is polar, you see? Molecule is polar. Okay. You can see the kind of positively charged hydrogen here. And this positively charged hydrogens and negatively charged oxygens interact with each other, as I already mentioned. And then these hydrogen bonds formed between hydrogen and oxygen of two different water molecules. That's important. You see two different water molecules. They define multiple properties, pretty unique properties of water. Okay. Hydrogen bonds are crucially important for living tissue. They can hold water molecules together. They hold DNA strands together. They determine the shape of the proteins. Now, what are other features of water that are determined by hydrogen bonds? Well, water is liquid at the room temperature precisely because of the hydrogen bonds. Water is cohesive and adhesive, meaning that it can stick to the surface and kind of crawl up. You can see it in the capillary effect. If you will take a porous paper and stick it in a bowl of water, water will move up against the gravity because of its adhesiveness and cohesiveness. That allows, for instance, water-based blood to run smoothly through the hydrophilic walls of blood vessels. Water has a high specific heat, which is very important because it means that it takes a lot of heat to change the water temperature, and that stabilizes the temperature of human body. It also has high heat of vaporization, which means it takes a lot of heat to evaporate water. We need a lot of heat to break down these hydrogen bonds, which means we can effectively cool our bodies by sweating the water out. And importantly, due to the unique features of hydrogen bonds, ice is lighter than water, so it will float on the water. And that means that when it's winter, ice on the surface of the bodies of water, like rivers and lakes, will insulate these rivers and lakes. They will not freeze all the way down to the bottom, and living organisms in the lakes and rivers will be able to survive. Another important feature that is crucial in terms of hydrogen bonds is that hydrogen bonds make water a good solvent for hydrophilic substances. So polar substances like sodium chloride or hydrophilic substances like glucose will form electromagnetic bonds with water molecules. You can see that negatively charged chloride forms electromagnetic bonds with partially positively charged hydrogen, and positively charged sodium forms electromagnetic bonds with partially negatively charged oxygen. On the other hand, hydrophobic substances like fats and oils cannot be dissolved in water. It fundamentally defines what can and cannot be dissolved in blood and bodily fluids, and how we transport hydrophilic versus hydrophobic substances.
So let's talk about van der Waals forces and pH scale, and then we move on to energy and large molecules, biological molecules. So van der Waals forces are weak forces that are formed between nonpolar molecules. For instance, you see a couple of examples here. 2 molecules of pentane with large surface area form strong van der Waals forces, while 2,2-dimethylbutane, a more spherical, weaker van der Waals forces. The example of van der Waals forces in nature, the gecko's feet can get attached to the smooth surfaces like glass by van der Waals forces. Now pH scale. So pH scale reflects the concentration of hydrogen ions in the solution, basically the acidity or alkalinity of the solution. So neutral pH is 7. Everything above 7 is considered to be basic or alkaline. Everything below 7 is considered to be acidic. Now, the pH scale is a logarithmic scale, which means every unit of the scale reflects a 10-time change in the concentration. So for instance, if you go from pH 3 to pH 4, it means that concentration of hydrogen ions goes down by a factor of 10. So essentially, the higher the pH, the lower the concentration of hydrogens. Okay. Now, we use pH scale, for instance, to identify problems with the blood. Blood pH should be 7.45, and for instance, in diabetes, due to the production of keto acids and ketones, blood pH decreases. It's called metabolic ketoacidosis. Case of respiratory abnormalities such as chronic obstructive pulmonary disease, the lack of proper ventilation prevents CO2 from being exhaled. CO2 accumulates in the blood, and that leads to respiratory acidosis. Now, in the human body, there are buffers. Buffers, the term buffer reflects the solution that can stabilize the pH by absorbing hydrogen ions or releasing hydrogen ions. So the most important buffer in the blood is carbonate-bicarbonate buffer. Here's the chemical reaction. When water combines with carbon dioxide, it forms carbonic acid, which then dissociates into hydrogen ions and bicarbonate. So when blood pH decreases, some sort of acidosis, bicarbonate here can absorb hydrogen ions. And if blood pH increases, then carbonic acid can release hydrogen ions. Now, we're practically done with elementary chemistry, and we're going to move on to the conversation about energy conversion in life. So the energy conversion in life starts with the solar energy. Virtually all life, directly or indirectly, is supplied to energy supplied by the sun. Plants convert the solar energy into the potential energy of chemical bonds. Okay, plants are considered to be producers, not consumers, producers. This is the reaction of photosynthesis, where carbon dioxide combines with water and produces, that's an example, glucose, well, carbohydrates, and oxygen is a byproduct. Animals are consumers. So they consume carbohydrates, they consume plants as the source of energy. Okay? And animals, they convert the energy of chemical bonds that plants formed to their own energy, to the ATP. Okay? They convert the energy of chemical bonds from plants into their own ATP. We can put ATP here, producing carbon dioxide and water. They basically oxidize glucose to carbon dioxide and water. Okay. And then when plants and animals die, then we have decomposers, various fungi, bacteria, various worms that break down the dead animal tissue, plant tissue, and release heat, carbon dioxide, and water as well. Now, let's say a few words about the reactions that occur in living organisms. Reactions in living organisms can be divided in anabolic and catabolic. Anabolic reactions are reactions of synthesis. Anabolic reactions form bonds, and that bond formation requires energy. So essentially, the product of anabolic reaction stores energy, but since it stores energy, these reactions essentially require energy, and thus they are called endergonic reactions. Examples of such reactions is the synthesis of triglycerides, the fat, or synthesis of carbohydrates or proteins. Reactions of breakdown here are catabolic reactions. The energy that is stored in the bonds is released, which means catabolic reactions are usually energy-producing, exergonic. When we break down proteins or fat or carbohydrates for energy, we essentially perform catabolic reactions. Now, how do we know that a certain compound is organic or inorganic? So in organic, in space, organic compound, carbon has a 4-valent, 4 bonds, and these bonds are oriented in tetrahedral configuration. The advantage of this is that it allows to form molecules with multiple functional groups, such as hydroxyl, and form chains. The almost infinite variety of structures of organic compounds explains how they can perform almost infinite variety of functions. And we further going to talk about 4 main categories of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Before we move on to those categories, we're going to say a few words about ATP. Okay? So, ATP is the universal energy currency in human and animal cells. When we consume food, we consume fats and carbohydrates, and we break those molecules down, and then we can store the energy that is released as the result of breakdown of fats and carbohydrates in 2 forms. It can be short-term energy storage, which is ATP, and long-term energy storage. So long-term energy storage for sort of prolonged, like basically your body's pantry, are glycogen in muscles and liver and triglycerides. Triglycerides are fats. They are packaged in adipocytes in your fat cells. They are not, glycogen and triglycerides are not readily available. They have to be converted through a series of chemical reactions into a molecule of ATP, which is a short-term energy storage. It is instantly available. Here's the structure of ATP. We call it ATP because it's adenosine triphosphate. Okay? And the energy is stored specifically in the phosphate bonds in the ATP, in these bonds between these phosphate groups. Okay? Most and foremost, it's the bond between gamma and beta phosphate group. So when ATP is synthesized here, by acquisition of solar energy by plants or chemical energy by animals, it is a short-term energy storage, and it can be almost immediately broken down to ADP, adenosine diphosphate, which will have only 2 phosphate residues and inorganic phosphate. And that reaction of breakdown of ATP releases energy that is necessary for cellular processes. Okay, and this scheme nicely illustrates the energy flow in the cells. So glucose can be broken down, oxidized to carbon dioxide, water, heat, and the energy of that glucose oxidation is then used to produce ATP. So reaction of ATP synthesis is essentially endergonic anabolic. And then ATP is broken down to adipine phosphate, reaction which is catabolic, reaction of breakdown, and exergonic, so it releases energy, which then can be used to form bonds between individual glucose molecules to form some kind of polysaccharide, for instance, glycogen. First biological macromolecule that we're going to be talking about are carbohydrates. These molecules consist of carbon, hydrogen, and oxygen atoms. Here's the empiric formula of a typical carbohydrates. The elementary building block of carbohydrates is a monosaccharide. So for example, there are individual sugars, one molecule, like a glucose or fructose or galactose are the examples of monosaccharides. When 2 monosaccharides, for instance glucose and glucose, combine, they form a disaccharide. Here it's maltose. Glucose and fructose will give you sucrose, which is basically a sugar. When many monosaccharides combine, there are, you can call them oligosaccharides, quite a few. If there are many monosaccharides, you get a polysaccharide, such as amylose or glycogen or amylopectin. So these polysaccharides, as you can see, are formed in the reaction of dehydration, and conversely they can be lysed in the reaction of hydrolysis. So both reactions of polysaccharide synthesis and polysaccharide hydrolysis are catalyzed by enzymes. So functionally, first and foremost, polysaccharides are energy storage molecules, such as glycogen, which is found in animal tissue, or starch and cellulose, that's starch, that are found in plants. Other functions include structural protein modification, and they also a part of the DNA and RNA molecules, so they indirectly participate in the storage of genetic information. Next, a biological macromolecule that we're going to be talking about are lipids, such as fatty acids, triglycerides, phospholipids, sterols, and hopanoids. So lipids are hydrophobic, meaning that they are not soluble in water. They consist mostly of the atoms of carbon and hydrogen, and many of lipids perform energy storing function. First lipid molecule that we're going to be talking about are fatty acids, which essentially long chains of carbons with attached hydrogens, the carboxyl functional group at the end. Fatty acids can be divided into 2 large groups: saturated, without any double bonds, and unsaturated fatty acids, such as this oleic acid or these omega-3 fatty acids, which have multiple double bonds, also unsaturated. This is mono unsaturated. These are poly unsaturated fatty acids. There are trans fats that you have probably heard about. So trans fats are chemically modified unsaturated fatty acids, shown here. It's a transoleic acid at the bottom. So unlike, contrary to the common idea, dietary saturated fats, such as stearic acid, they do not increase the risk of cardiovascular disease. But dietary trans fats that are found in processed foods and margarine, they increase the risk of heart disease. There is a hypothesis that polyunsaturated fatty acids, such as this omega-3 fatty acid at the bottom, may reduce inflammation, may reduce blood pressure, reduce clotting. They're not produced in the organism, and it's recommended to have them as a part of a healthy diet. When fatty acids, shown here, combine with a glycerol molecule, and that combination produces triglyceride, neutral fat. You can see that it is also a reaction of dehydration, and the main function of triglycerides is to store energy. If triglycerides contain unsaturated fatty acids, they are liquid at the room temperature. We call them oils. Those are plant triglycerides. Triglycerides that contain saturated fatty acids are solid at the room temperature, and we call them fats. Now here at the bottom, you can see a couple more examples of another type of lipid. It's basically a condensed polycyclic compound. Those are sterols and hopanoids. So sterol is on the left, hopanoid is on the right. Both sterols and hopanoids are found in the cellular membranes, and they regulate the fluidity of the cellular membranes. Sterols regulate the fluidity of eukaryotic cellular membranes and eukaryotic organisms such as humans. Hopanoids regulate the fluidity of cellular membranes in bacteria. Another role of sterols in a human organism is to serve as the precursor for various hormones, steroid hormones such as estrogen or testosterone or cortisol. Now, the last type of lipids that we're going to be talking about are phospholipids. They contain glycerol, just like triglycerides, two fatty acid residues, saturated and unsaturated, and phosphate residue right here. Glycerol and phosphate, highlighted by red, form a polar hydrophilic head of a molecule, and fatty acids, highlighted by yellow, form a hydrophobic nonpolar tail. These phospholipids form phospholipid bilayer, which forms cellular membrane. Here you can see an illustration. Hydrophilic heads in that bilayer face outward, the outside of the cell space, which is usually, hydrophilic, watery, and here they face, say, cellular cytoplasm, which is also watery. But in the middle here are nonpolar fatty acid tails. They face into the membrane, okay? And that hydrophobic layer of the membrane forms selectively permeable barrier. Now, next biological macromolecule will be a protein. Proteins consist of amino acids. There are 21 different amino acids. And here you can see examples of some amino acids that can be found in the proteins. You can see that they all share similar structural elements. They have amino group, carboxyl group, and a side chain. And they are different because different amino acids have different side chains. You can see side chain of serine is different from side chain of glutamine, for example. Essentially, side chains of amino acids will determine the structure and the properties of a protein. When two amino acids combine in the reaction of, again, dehydration, they form a peptide bond. And here you have a dipeptide. When many amino acids combine, they form a polypeptide shown here. And the sequence of amino acids in the polypeptide is called a primary structure of a protein. Now, these polypeptides start to fold, and they fold into what's called a secondary structure, which is comprised of two main structural elements, alpha helices and beta sheets. Alpha helices and beta sheets fold further, forming a tertiary structure of a protein, essentially a three-dimensional globule, okay, that is determined largely by the interactions of side chains of amino acids. Sometimes these globules are combining together, forming a quaternary structure. You can see light gray and dark gray globules, two globules. So basically it's a protein that consists of several polypeptides, several amino acid chains. Subunits of a protein are not bound covalently, so some kind of physical, not chemical interaction. What's important is that proteins can be denatured, so they can be structurally altered. Okay, quaternary structure can be disrupted, tertiary structure can be disrupted, and secondary structure can be disrupted. Denaturation, i.e. structural alteration, will lead to the functional changes, inability of a protein to perform its function. Moreover, if you will replace one amino acid in the polypeptide, it may fold differently, and that single amino acid change can alter the protein structure and therefore the protein function. Now, last bit about biological macromolecules will concern the nucleic acids. So DNA, shown here, is a double-stranded helical molecule found in the nucleus. Its function is a storage of genetic information. It encodes proteins, regulates various physiological processes, while RNA, single-stranded molecule shown here, is a messenger molecule and serves as the template for protein synthesis. It essentially conveys the information from DNA to ribosomes for the protein synthesis. Nucleic acids consist of nucleotides. DNA consists of 4 nucleotides: A, G, T, and C, adenine, guanine, thymine, cytosine. RNA, instead of thymine, contains uracil, so A, G, C, and U. The nucleotides form complementary pairs. In DNA, it's AT and GC. In RNA, it's AU and GC. The main difference between RNA and DNA nucleotides, in addition, it's not T but U, is that in DNA, nucleotide contains deoxyribose sugar. And in the RNA, it contains ribose sugar. Now, as I mentioned before, DNA is the double-stranded helical molecule and RNA is a single-stranded molecule. Double-stranded molecule means that these two strands are complementary to each other. So where you can find A, in the opposite strand there will be T, G in the opposite strand there will be C, which means that sequence of one strand defines the sequence of another strand, which means that one strand serves as the backup for another. So the last bit that we're going to be talking about are enzymes. Enzymes are catalytic molecules that speed up the rate of a chemical reaction in the cell. Every reaction has to go through that bump that you can see on the graph. It's called activation energy. So enzymes reduce activation energy, making the reaction go faster. Importantly, that enzymes do not change the ratio of the products or the yield of the products. They just make this chemical travel from point A to point B faster. In the course of enzymatic reaction, the substrate, a chemical in the beginning, binds the active side of the enzyme. That's the side where this reaction happens. Enzyme changes its shape and weakens the chemical bonds in the substrate, and then substrate subsequently is converted into products. It doesn't necessarily have to be the breakdown reaction that is shown here. It can be a reaction of synthesis or something else. Important feature of the enzymes is that they are, first, absolutely fantastically specific. They are not wasted in the reaction. So you see here that enzyme comes out of the reaction unchanged. And their specificity is due to that lock and key match between the substrate and the active side of the enzyme. Every enzyme has optimal conditions to function. For instance, enzymes in your stomach will have low pH being optimal. And enzymes that work in your gut will have neutral pHs being optimal. Different temperatures, different concentrations of enzymes, substrates, and other chemicals, all of it contributes to how well the enzymes work.